Hello, Jesse. Ujjwal. Hello, Mayukh.
Hello, everyone.
Hello. Hello, Dick. How are you?
Pretty good.
All right. So we've got probably enough people here to start. Welcome to the meeting. Hope everyone's doing well. And let's see. We have a couple things on the agenda today. One is the Dick had an idea for the Laplacian embryo, which we'll talk about. And then we need to talk about the GSoC final reports and sort of the last week of that. And then maybe we'll talk about I'm preparing a blog post on DevoLearn, and so maybe we'll talk about that as well. I think it's probably okay, but I wanted to go over it a little bit. So I guess we'll start with the Laplacian embryo idea.
Okay. Well,
yeah.
Yeah, well, basically there are two methods of doing fluid mechanics. And that's where you're trying to calculate the flow of a fluid. And one way is to divide space into little boxes and calculate how much fluid comes into each box and how much goes out. And that's called Eulerian, I think it's Lagrangian, not sure, maybe it's called Eulerian mechanics. The other approach is to let the box move in space and follow the matter in the box. And that's Lagrangian mechanics. Okay, now Lagrangian mechanics is what's appropriate for embryos because cells move in space inside an embryo. Okay?
Yeah.
And they move and divide, and since we're getting a lot of information on the motion of smooth cells and the orientation of cell divisions, I think it's perhaps time to start taking a Lagrangian point of view of how an embryo develops and see if we can calculate the development of an embryo and see how far we can get with that. But also deliberately introduce errors, for instance, errors in angle division, errors in cell size, etc., to see how stable development is. Because this is one of the remarkable things of development, and that is that you end up with a whole organism despite its the variability and the behavior of the cells. So these are the ideas then. I sent 3 papers, 2 recent ones on nematodes, I think they were nematodes, and a paper I did a while ago on the motion, what happens in an embryo at the very first cell stage at low gravity, which was with some fellows from the Netherlands who are experts in Lagrangian mechanics. Okay? So I figured those 3 papers, if somebody's interested, would get people started on the reading, and then we could see what's available in our databases now that might make this practical. Okay.
Sounds good. Yeah.
Okay, so there's been for decades there has been the idea that one can calculate an embryo, and I think it's time to test that idea. Okay.
Yeah.
Of course, I don't know if it'll actually crawl out of the computer, but.
Well, I don't think anyone's done that. Yeah. Kind of like the hype around OpenWorm, like people always talk about, like, well, crawl out of the computer. Well, the worm. So.
Right. Anyway, that's the idea. Now, if it grabs somebody, then maybe we can proceed with it. But it's obviously a large combination of a database plus plus simulation of fluid motion, basically, in Lagrangian mechanics. Now, Lagrangian mechanics is used in water research, for instance, in the making of a wave, where you want to follow the drops in the wave and see how they move, and they can separate from the mass. Whereas in standard mechanics, you can't let things fall apart. Okay.
Yeah.
So Eulerian is excellent in that sense, that it allows calculations that are not possible in the standard approach. Okay.
Yeah. So that's like finite element analysis.
Yeah, except the finite elements move.
Oh, okay. Yeah.
Okay. You know, they move, they can generate forces between themselves, and they can separate, and they can join, and things like that. Okay.
Yeah. Susan says ComSol physics is Eulerian, so ComSol is the multi-physics modeling platform.
Yeah, there is. Lagrangian mechanics is not very common. Okay. Yeah, ComSol does. Yeah, so they still don't have it in there. And it's hard to do it right. I mean, the guys in the Netherlands are experts at it, and so reading their papers and seeing if we can get their software again. I mean, it's been it's been quite a while since I worked with them. And they probably improved their software quite a bit. I don't know if the current versions are available or not. But, you know, first the question is interest.
Right.
Okay. But this question of calculating the embryo has been a challenge in the literature for a long, long time. And now I think we're reaching a point where we can seriously think about it. Now, you see some of the data which we might be, can we get things like the orientation of a cell during development?
The orientation in what way? Like the...
In space, which direction? If you put an axis between the two centroids of a cell that's just divided,
yeah,
that's its orientation in space.
Right.
Is there any data available on that orientation?
I'm not sure. I think it would just have to be a matter of like tracking the, like if you had a good resolution image and you could track it in the way that, well, they have tracking software that is sort of allows you to sort of calculate space but then we also have our feature detection, so we might be able to detect features that sort of align with that. So in other words, if you have a cell dividing, can you detect that as a feature, and then can you detect the variation in that feature? Like, what is the, you're going to use a couple of landmarks and then maybe make a calculation from that.
Okay. Okay, so the question is, if we can get that kind of data and its variability, then we can try to use that as a local constraint in a simulation. Okay.
Yeah. Yeah. Let's see. Susan said, OpenFOAM is another platform. So I guess for simulating physics.
Cool. Okay. Okay. So Susan, you've delved into this question of Eulerian versus Lagrangian mechanics. Okay, don't hear from her.
Okay. She says yes a bit.
Okay, fine. Okay, well, if anyone can do this, I think it's this group that can get into it.
Yeah. So yeah, so you sent me some papers as well, and yeah, we can go over the papers.
Yeah, anybody's interested. If you could post them for the whole group, then we'll see who's interested. Okay.
Okay. All right, let's see. Yeah, Susan has more familiarity with Lagrangian methods, I guess. So here's the drive folder with the papers in it. I've pulled them up.
Okay.
And let me share my screen. And I will go through these quickly to see what's in the folder, and then if you want to follow up on it, we have this folder here. If you need permissions, let me know. So the first one here is Wittgenstein and Strunz and Weiss. This is the group from Germany, I take it. And so this is mechanical cues in the early embryogenesis of C. elegans. And so this is where biochemical signaling pathways and developmental processes have been extensively studied, yet the role of mechanical cues during embryogenesis is much less explored. Here we have used selective plane illumination microscopy in combination with simple mechanical models to quantify and rationalize cell motion. So they're doing a similar thing to the cell tracking with, microscopy images where you have a movie. And then as a result, we find that cell organization in the embryo until gastrulation is well described by a purely mechanical model that predicts cells to assume positions in which they face least repulsive interactions from other cells and from the embryo's eggshell. So there's, of course, we don't talk a lot about the different phases of development in this group too much, but this is like early embryogenesis. This is I don't know how many, I can't remember how many cells you have to get to gastrulation in C. elegans, but the idea is that they divide from a single cell. They divide into two cells. One is the posterior end, one is the anterior end, and then those two cells form two what they call sublineages. So the AB sublineage and then the P sublineage, and the P gives rise to a lot of the specialized lineages like MS and C and E, and those are usually like different tissues. P, if you follow that, there is the germline, which is where the eggs come from. So we have this very early organization, but then they're saying that if you look before gastrulation, which is sort of like the first sort of folding of the embryo where it kind of starts to take a little bit more shape than just a sphere, that this is all mechanical in nature. And so let's see if they have any images in it. So this is the setup here. So they're doing this custom-made SPIM image acquisition. There's image processing, and then this is let's see. This is the different sections of C. elegans that they have, and actually, looks like they have a graph. Looks like something Mayukh made a couple weeks ago, where you have this three-dimensional space. You have these cells, and you put a reference frame around these points, and then you track the cells, and you find that they have this trajectory. Yeah, Mayukh made a figure like this a couple weeks ago, and I think it's in one of his notebooks, where he was tracking sort of the position of the cells or the cell lineage in the embryo. Given that you normalize the embryo across embryos and across divisions, you end up with this kind of space, and then you can track the motion of these cells. And these motions, of course, are, some of them, they're maybe driven by mechanics, but it's basic cell motility. The cells have to move when they divide. When they divide into two cells, the cells reposition themselves. So they track nuclei. So again, this is very similar to what we're doing in the machine learning context, where you're trying to find like a centroid or something, and then you have this. You can track those centroids, assign them a position in the reference. Assign a reference frame and positions, and then that's what we have. So then they've done this. What else do they have in here? So they have these cell trajectories. Way it works, so you can build this sort of model of spheres that explain, the model of the embryo, it looks like, and then you can actually model it with different forces and use that. So I mean that's what this paper looks like. It's something similar to what we've been the direction we've been going in with the machine learning. It seems like there might be maybe another level we need to add to it, where you have like a physics model, where you have the information about the positions or the tracking of the cells. Now you need to have like sort of a more general framework of how the cells are behaving in some way. And so they're arguing that a good physics model will do this. So we probe the cell arrangements that arise as early in C. elegans embryos without the guidance of an eggshell. Experimentally, a T-like arrangement of ABA, ABP, EMS, and P2 have been observed when removing the eggshell. So this is actually an experiment that we did. I don't know if I was talking about with this group maybe several years ago, but there's a set of experiments that you can do that when you take the cells out of their egg and you dissociate the AB and the P1 cells, and you put them in a culture, you have like a plate that has like culture media on it. The cells will start dividing, but they won't divide in the way they divide in the egg. They actually divide into these barbells, and they form these like chains, like long chains, and they just kind of line up like end to end, and then maybe form a barbell at the end. And so I don't know, like those papers are like 40 years old, but they're interesting in the sense that it suggests that there's something going on in the egg that's different from what's going on in the, if you take the cells out and you just say, well, there's no egg anymore, can they use the same sort of cues that they're using in the egg? And the answer, at least based on that set of experiments, is no. And so that's how the physics layer is important here. Yes. So then that's that paper, and then we have the Tian paper, why and how the nematode's early embryogenesis can be precise and robust. So this is a physical biology journal, and this is the early embryogenesis in the nematode C. elegans is well known for its stereotypic precision of cell arrangements in their lineage relationship. So that means you can calculate a cell lineage tree, and it's the same in every embryo except for like a mutant, and it happens the same way for every egg that forms into a worm. And so, but however, the origin of the robustness in the cell arrangements is poorly understood. Here we set out to provide a mechanistic explanation of how combining mechanical forces with the order and orientation of cell division ensures a robust arrangement of cells. So they use a simplified mechanical model. They simulate the arrangement of these cells. They introduce different disturbances. So they're ordering simultaneously. Oh, is that go back up? Okay. Was there something you wanted to point out, or?
No, he was talking to me.
Oh, That's all my way, sorry.
Thanks. So they do these perturbations, ordering, simultaneity, and the division orientation of cell divisions. So they're talking about that orientation of divisions. And then our work provides insights into the developmental strategy and contributes to the understanding of how robust or variable the cell arrangement can be. And so this is basically where they want to basically perturb the egg, or the cells in the egg, to see if they form different patterns. If you do something like, I don't know, introduce different temperatures or introduce different forces. And so that's what this paper is about. And so this actually gives us, in modeling terms, sort of a parameterization of this regularity. And so they talk about, again, they review the mechanical forces. And so you see the egg here in the cells, and they have these mechanical forces that act between cells and then between the eggshell and the cell. And then they show the classic lineage tree, which is just how things are ordered in time. Well, they're ordered in time, but they're also ordered along this anterior-posterior axis. So the C. elegans lineage tree at least has this order in space. It's one-dimensional, but that's how it's laid out. So that's that. C is, okay, this graph C is the relationship between spring-like repulsive force and cell-cell distance. So you have this force that's generated, and then this distance, and I guess the idea is that there's this relationship between forces between the cells and the distance between cells. And so they model that as well. So let's see. They model this. They focus on the mechanical robustness of cell arrangement against perturbation up to the 24-cell stage in C. elegans. So it's not very many cells that they look at. They look at like a pretty early stage of development, but it gives you some idea of basically what's happening when you get these sublineages formed. So by the 24-cell stage, you get all your major sublineages formed. When you get to the 8-cell stage of C. elegans, they have what they call the 8 sublineages, which will be the sublineages that exist for the rest of C. elegans' life. There are 4 AB sublineages, and then there are 4 sublineages in the P sublineage. So there's I can't remember the names right now, but but they're 4 and 4. So there are 4 that come from the posterior 2-cell and 4 that come from the anterior 2-cell. So there's that sort of symmetry. And so then from there, that like all these sublineages are sort of in maybe some position in the egg, and you can look at basically how that establishing process works. So maybe it makes sense to look only to the 24-cell stage, because that's where a lot of your sublineages are sort of positioned to start to proliferate in sort of the areas of this embryo that they're going to later form tissues and other things. So let's see. So this is the figure here. So this is simulation of perturbation of division ordering. Repermutation for 2 division events. Cell-cell contact map of 8-cell stages, which is A. So this is, I guess this is where they do a normal order, a reverse order, and then a simultaneous. So they have these different conditions that they do. And then B is this one, cell-cell contact map of 8-cell stages after changing the sequence of events 2 and 3. So they change the event order and they see what the contact looks like. So they're like, you're taking that lineage tree, they're changing the division events, or the order of them, and then they're looking at these cells. So I guess what they're doing is they're taking, if you didn't divide a cell, and you divided some other cell and you wanted to look at how they would sit together. They're basically asking, do these mechanical constraints explain the order of the timing of division and their relative locations? Or is it maybe, doesn't really matter because, the cells are basically all in the same place where that, it's all equivalent and there's just this general set of forces. I think that's what that's kind of what they're arguing here. C is cell-cell contact map of 12 cell stages after synchronizing events 3 and 4. So again, they play around with the timing of cell division, and they look at the contact between cells. And then D is the cell-cell contact map of the 24 cell stage after reversing the sequence events 6 and 7. So you see that there's this contact map. And so that's an interesting set of experiments. I think, there's a lot to be learned from that, about playing around with the order and thinking about, like, why is C. elegans development, why does it unfold in a certain way? We just kind of sometimes default to the explanation about genes and this and that, but, it could be mechanical constraints, or it could be something else entirely. But I think doing these kind of experiments are quite useful for that. And you can do this, I think, computationally if you have like a good model of the cells. And this is something that can be done just with some computational experiments. So they do the simulation of perturbation of division orientation. So this is where they're looking at division orientations. And I think we have some data on that. I don't know how accurate they are. Those were derived from the EPIC data set a long time ago. So we might pull those back up and look at them. Again, I don't know exactly how accurate they are, but they might be useful to look at. We might be able to re-extract them out of the image data, is what I'm saying. And that might be a bit more accurate. But so they've done this experiment. And again, this is just like tracking the mother cell and the daughter cells, their centroids, and then looking at the angle of, so one daughter cell goes in one direction, one daughter cell goes in another direction. And there's this angle between them. And so the angle then, what does that represent? Is that like, is that random? Is it something that like is produced because of like pure mechanical constraints? Or is there something going on in the lineages where they divide at a certain angle? And that's the sort of thing you would ask there. And so they kind of they ask this question and answer it a little bit. So if you're interested, look at that. And then quantification of reproducible final 8-cell pattern in 6 division sequences. So this looks like they're just kind of reconstructing the different lineages and looking at their... they're looking at normal-handedness and reverse-handedness. So they're actually flipping the embryo around, and they're looking at, like, if you flip the posterior end to the anterior end, right? Like, or you're flipping the left side to the right-hand side. What is the consequence of that? So, yeah, that's something else we might look at. So that's that paper. And then the third paper is this Nouri and Rowley paper, which is Rowley instability of the inverted one-cell amphibian embryo. So this is Nouri. This is on the Rowley instability. And this is another physical biology paper. The one-cell amphibian embryo is modeled as a rigid spherical shell containing equal volumes of 2 immiscible fluids with different densities and viscosities, and a surface tension between them. The fluids represent denser yolk in the bottom hemisphere and a clear cytoplasm and the germinal vesicle in the top hemisphere. And so the unstable equilibrium configuration of the inverted system depends on the value of the contact angle. So this must be what you were talking about, Dick, when you were talking about the Lagrangian methods, sort of moving towards that.
Yeah, this paper depended on Lagrangian methods, as I recall.
Okay, yeah. And so the two dominant types of modes of perturbation give rise to axisymmetric and asymmetric sloshing of the cytoplasm of the inverted embryos, respectively. This work quantifies our hypothesis that the axisymmetric mode corresponds to the failure of development, and the asymmetric sloshing mode corresponds to development proceeding normally, but with reversed pigmentation for inverted embryos. So here they're showing like some of these properties of the cell, and they're looking at like these instabilities. So Rayleigh instability is, I can't quite remember what that is.
Oh, that's if you have a cylindrical piece of fluid, it breaks up into drops.
Oh, okay. So this is, yeah, they have all the notation up at the top of the paper, so that's always good. And then a schematic diagram of the unstable equilibrium of a sphere filled equally with two immiscible liquids, and with the heavy one on top, and when the contact angle is less than 90 degrees. And then B, when it is more than 90 degrees. The dotted line shows the flat surface when the contact angle is at 90 degrees. So you get this slight curvature. And then, so this is actually inspired by amphibian eggs. So our axolotl eggs that we've talked about in the meeting are amphibian eggs. And so this is a bit different from C. elegans. We just talked about C. elegans in the last two papers. This is, of course, a different system, and so there's a different sort of mode of development here. And so we have this. We look at like what they're doing is they're looking at a fertilized egg, which we can think of as a one-cell amphibian embryo. When this type of one-cell embryo is inverted prior to the first cell division, it either develops with pigmentation of dorsal and ventral skin reversed, or does not develop at all past gastrulation. So now this embryo, this type of embryo that we're talking about here, is different from C. elegans, and that it doesn't have that same programmed fate aspect to it. There's a lot of signaling that goes on, and you have different things. The egg is much different in terms of how it develops.
Yeah, I put that in mostly to give an example of applying Lagrangian mechanics in embryogenesis.
Okay.
Okay?
All right.
And I don't think anyone else has done anything similar.
Yeah. So we divide the problem into two separate steps. First, we calculate the unstable equilibrium configuration of the system, where the heavier liquid is on top, and try to find the equation describing the non-planar interfacial surface between the two liquids. And then the second thing is that they introduce some small deviation from the equilibrium configuration and study the growth rate of each normal mode of perturbation. This is a Rayleigh type analysis after Lord Raleigh's instability analysis of a long cylindrical liquid. This will show the likelihood of any type of flow of the heavier liquid down towards a stable configuration in which it finally settles at the bottom. So that's what they're doing with this. And so they use, this sort of mathematical model. So you're using a set of Navier-Stokes equations for free surface flows. And so that's a simulation you can run in a computer program, of course. And then you can get, you can solve some of this. So let's go through this. Here's a nice visualization of what they did. So visualization of different modes of excitation of the interfacial surface when the heavy fluid is on the top half of the sphere. The numbers underneath each mode shows the M and N parameters for each mode. This figure only shows the heavy fluid on top and is a colorful only for visual convenience. So the colors don't really correspond to anything necessarily. But this is just a visualization of the different modes of excitation. And then, let's see what else is useful here for a quick overview. So this is an example of the oscillation of modes. And so for each simulation, the system is started with the interfacial surface to be one of the modes with the heavy fluid at the bottom. And then at a later time, the interfacial surface is expanded in terms of all the modes, and the coefficient of each mode is calculated. So each plot shows these coefficients versus time. So this is time, and this is the coefficient value, indicating that to a fairly good approximation, only the initial mode is oscillating, and the rest of the modes stay calm, showing that the modes are in fact normal modes of the system. So this is for the different modes. And then this is a velocity field analysis. So this is a way to look at fluids, look at their motion and the velocity within that area, within the fluid. So this is a velocity field. You have these arrows that are curved and different weights, as you can see, and this describes some of the results of their simulations as well. So I think I'm going to wrap that up. I think that's a nice set of papers. And again, if you're interested in this, go to that folder, look at the papers, and share some of your ideas with us. Let's see what we have in the chat here. We have some things. So we have, I'm interested in fluid modeling for my thesis, Susan says. And then Mayukh says, in fact, this is possible in DevoLearn. So yeah, we've talked about how this is consistent with what we have in DevoLearn. Here's an expert on Lagrangian Mechanics. So if you're interested, Veldman. Dr. Veldman recommended OpenFOAM to me. And that's Susan. And then Dick says, can we say anything about variability at the local level, such as spatial angle of a division versus longer range effects? That's a good question. If we have the angles and we know, like in C. elegans, we know what the cells are that are dividing in the angle, we might be able to make some statements. In fact, I think we do have like an analysis of the angles, like I said, and I can't remember what that involved. I don't think it ever got put into any paper or anything, but I'll have to pull them up for next time and see what they look like. So I don't know if they really describe longer range effects. They very well may, given that we saw in the papers that there's something going on within the first 24 cells. There's like establishing some sort of spatial relationship that's going to be important later in the embryo, but we don't know. And then this is the link to OpenFOAM. So yeah, check that out. So next, let's go on to the GSoC projects. So a couple words before we start with like updates. Number one is that this is the final week of GSoC. So everyone, Ujjwal and Mayukh are probably excited to be done with GSoC. And if we, I think I can't remember exactly the time that they're due. I think they're due this weekend or next Monday or something. The 31st, is it? I would double check the schedule just to make sure, because they have like a certain time of day it has to be in by.
It's 31st next Monday.
Okay, yeah. And so, and the time, of course, is UTC, so make sure that you convert the time so you have it in on time. Then, I guess what you need to turn in, like I said last time, is some sort of README, or, you could use a gist. I don't know what you want to use to submit. Okay. But, just basically have a description of the project. And this is, irrespective of if you have like a bunch of notebooks. You basically want to clear, like, sort of README that says, this is what I did, this is what I learned, and then these are the links that are important to see the project. And again, they're going to go into the project, they're going to go into this README, and they're going to click on the links, and they have to run or produce some sort of output in order for you to pass. So, I mean, like, it's not to scare you, it's just to say that, like, you just want to make sure that they're all sort of in working order, no bugs or anything. And I just say that because, if something ever happens, people get upset or something. But So then I think both of you have done pretty good work. I've asked Ujjwal to assemble all the links to the work that he's done. So he's got a bunch of links in different places. I wanted to make sure that we had a good record of where those were. And I'm sure that'll be good for the report. And then Mayukh has, of course, pushed a lot of things to DevoLearn, the organizational repo. So again, this is DevoLearn. Let me share my screen. I don't think Dick has seen the DevoLearn organization, but this is DevoLearn and has a number of things. So we have data science demos, which are actually something that Mayukh and Krishna Katyal have contributed to already. So the network stuff is from Mayukh, and he pushed a bunch of things related to these embryo networks. So he's produced some graphs here that are, kind of show some principles. There's some data in here and some other animations. And then we have Krishna actually pushed a tutorial on Unix command line basics or Linux command line basics. And so that's nice. It's a notebook. And I see that in Slack, Mayukh suggested, making a notebook free version, which I agree with, because I think notebooks on GitHub are not always, they don't always render properly. So, but we have, this is C. Elegans DevoLearn. This is the library that Mayukh has been working on. Well, actually, no, this is Mayukh's DevoLearn here. This is the one that Mayukh's been working on, the libraries with all of the notebooks. And then C. Elegans DevoLearn is actually Ujjwal's project, and that's with a lot of the things pushed to this. And so we have, so that's all in place pretty much. And then the contribution guidelines, which Ujjwal and Mayukh have, I think Ujjwal has contributed to mainly. And this just describes like how do you contribute to this. And then the final one is the media, which is just where we put a lot of files that don't really have any other home. And so we're going to publicize this. I think starting this week, we're going to sort of announce it to the world. And so I've been preparing a blog post. So this is my blog, the inner guts of my blog here. This is where I'm sort of editing the thing as we speak. Well, not right now, but after the meeting. So I've got some images here. I've got some description. I mention everyone, kind of like how it came to be. This is just kind of describes a little bit about what it is. And then we have some images from the PyP project description, the GitHub repository, and then just kind of like finishing up with this, talking about DevoZoo a little bit, and then just letting people know that they can contribute. And so I think that's a good, I'll put that blog post out sometime this week, maybe tomorrow, and then we can start to announce it. And then we'll turn the GSoC projects in by the 31st. And then, and then we can, if we want to build on top of this, and I think it's nice to have as a reference, as a resource, but I think we can also build on top of it, like we just talked about with the physics modeling. It allows you to, this platform will allow you to take data or take images or movie images, movies of images, put them into this program, and it will yield some sort of quantitative analysis of those images. So it could be like cell centroids, it could be other types of information about angles or about networks between dependencies between the cells in terms of distance. So we have all of that in place, and that can be done. And now the next step, of course, is to use, like, maybe build a theoretical model or some sort of other model that, whatever we want to do. It's going to be a lot easier now. And people in the community can use it as well. So if people want to, if someone is interested in the topic and they don't know where to start with respect to, quantifying their images, we have a platform for them. And so, I think this will be an exciting thing to see, kind of get used. It was exciting to see it built. So thank you, Ujjwal and Mayukh, for your efforts this summer. And now I'd like to have you give updates. And next week we're going to do some presentations. So I wanted maybe if you could do like a 15-minute presentation in PowerPoint, kind of to recap everything so that we can have them for people so they can see the sort of a presentation of what this is as well. We'll just do it in the meeting in PowerPoint, and then we can have it recorded, and then we can go back to it for future reference. So who wants to go first for their update?
Yeah, I can go first.
Okay.
So I'll just share my screen.
Okay.
Hold on a second, I'll just...
All right.
So... Can you see my screen?
Yes.
Yeah. Can you see it now?
Yeah, I can see it.
Because I can't see it myself anyways. So, you guys can see the Travis CI page, right?
Yes.
Like, so what I did last week was that it would be easy to assume that in the future, like in the very near future, there will be more contributors who will be coming to GitHub and coming to DevoLearn to contribute and to make it better, right?
Yes.
So for that, what I did was that I set up unit tests. So what these unit tests will do that they will make sure whether or not the whole code base works after their pull request, or in fact before their pull request is merged. So we can just automate this test with everything is automated now. So whenever someone makes a pull request which basically breaks the code, it will show us a warning right there. So we can just warn the contributor that please make these and these changes to not break it. So so I just set up these tests, and then there's this badge on wait. So can you guys see there's this badge on GitHub now on DevoLearn. This badge called build passing. So whenever we see, if the build is failing in any case someday, then we realize that something within the code base is failing. If something fails, then we can just run the test again and check the logs and see where it's failing. So that will be a convenience later on. We have to maintain this project later on, right? So this will make it way easier. When more contributors come, and I assume more people will come soon enough. So yeah, that's pretty much it. I couldn't do much.
Okay.
Everything's pretty much done. GSoC stuff is kind of done.
Yeah.
Let me share my screen. So this is something that we have discussed. Can you hear me? Am I audible?
Yeah, I can hear you. I can see your screen coming up.
So I have can you see my screen?
Yes.
So basically what I have done last week is we have decided to integrate the Travis CI, and I am working on the Circus CI thing, so it will be done pretty soon. Apart from that, I have worked on the DevoWorm GitHub IO. So this is the DevoZoo that I'm talking about. So it is now completed. I have given a broad overview of what kind of data it is. For the Excel files, there is the Excel logo, so that people can think about there is the Embryo data and initially which has an Excel file, so it may be CSV or Excel file. Then the remote microscopy videos. So these are the links. If you click on these links, you will be redirected to the shareable drive. That's why I'm using incognito mode, so that it is just to make sure that it is freely available. You don't have to ask for permission. So these clicking into you. So the person who is reaching a portal can easily see here itself like what kind of microscopic videos are we providing. So he can either like view in the I don't know why it is not playing. So yeah, basically like it should like you can see here itself like what kind of microscopic data we are giving. I think like it might be problem with my internet or something. So this is the thing. You can see it there, and like all the videos, then there is different data sets that have been provided by DevoWorm. So I have listed them all, and some research papers like the listed naval papers like on the research gate. So these are the things. After that, like just to give a little overview, like what we do and what are the latest going on, I have embedded the tweet feed of OpenWorm and Orthogonal Lab. And like if we ever set up an account for DevoLearn, so I have tried now left this space vacant so that it can also be there. So this is the next thing. After that is just the mission and design of OpenWorm, and there is the microscopic tutorials. Then it is like it's the DevoWorm could maintain the website. So if you want to contact them, you can contact them. This is the basically DevoWorm thing. You can visit the repo here. After that, in the yeah, so with the same with the DevoWorm AI, if I can just quickly open it. So in the DevoWorm method, a person can go and click here, and he can again visit to the DevoWorm.
Ujjwal, I think you're disconnected for a minute.
Yeah, I guess he got disconnected.
Yeah, he'll be back in a minute, I think. Yeah, I mean that's pretty much. I mean, he'll be back. He'll finish up, but I think that's the overview of what he's going to have. I guess it's live now, so I can send out. Well, I think I'll put the links in the blog post, and I can send the links out as well to people if they didn't get the. In fact, so we have the data that's organized. The data sets are organized, that ones that we had that were sort of collected from different sources. So we have a broad variety of input data, and those are organized. So people, they come into the thing and they want to do some analysis. They want to find a data set that they can, quantify really quickly. They can download a video, and then they can put it, up into this platform, and then it'll give them some quantitative output, and then they can work with that output, and it will give you, a pretty quick analysis of this. I think a lot of the analyses in the past have been sort of, people, like you saw in that paper where they had a method section where it was very, it basically described what they were doing, but then could you do it yourself? That's why I think one of the reasons why we went into this whole project is that, we wanted to make this a lot easier to do for a wide variety of different species or whatever. So, here's Ujjwal, he's back. Hi, Ujjwal.
Sorry, I don't know how I got disconnected. I am giving presentation to myself.
Yeah.
So continue once again.
All right. Why don't you share from where you were?
So where I disconnected? What was the last thing I'm talking about?
You were on the DevoWorm AI page.
Oh, so yeah, so DevoWorm AI page. So as you can see, like I don't know if I have stated it or not. So like DevoWorm is also connected from here itself. So you can visit DevoWorm from here. Apart from that, like these two things, I have started working on two notebooks. Like I don't know where the Krishna Patyal is on his progress for the general image segmentation model that we have proposed in the DevoWorm AI. So I have started working on it anyways, and like I have set up two column notebooks right now for the mask RCNN and the image segmentation. So in the image segmentation by TensorFlow, it is not completed yet, but I am trying to give as many as possible as I can so that people can really, if you want to understand like how the things work, they can really get into it as deep as they want. So I'm trying to build that notebook. It will be complete by tonight. And the mass RCNN notebook, so it is basically like people of don't want to, like, sometimes when the researcher is doing some image segmentation, he don't want to get deep into what mass RCNN is doing. He just want to get his study done. Like if a biologist is doing something, so he don't want to like really like go deep into what mass RCNN. He just want to get his segmentation done so that he can again focus on his biological domain. So what this code does is it provides a simple, it is not editable for them, like I will just provide them the editable link for the image upload, and they can simply like do segmentation of the images, and like there are some parameters that we can adjust through sliders and these things. So I think like this will provide a benefit to the community, because like people can easily use the models for their purpose. So these two things I am working on, and apart from that, I have like started working on Devo editor that I have proposed. So it is almost complete. Like I have to just do some JavaScript tweaks. Like it is not cropping yet. So these sort of things will also be completed by this week. So I think that by Wednesday, I will host these two notebooks on the Devo node, on the like DevoLearn repository, which is for data science. I will upload it to that tutorial, and I will connect the DevoWorm AI with the Devo editor so that people can use it. And I will also like trying to make a Flask tab for general segmentation so that people can upload their microscopic image and get segmentation quick. It is not based on for particular like C.elegant segmentation. It is based for more general purpose. So these three things are the things that I'm aiming to get completed by Wednesday itself. And I have also completed my blog post, and I'm a little behind on the blog post. So I have completed it. It's just a matter of time. I will upload all of them. And I have prepared two tutorials in the introduction to CSS and like some JavaScript basic tutorials, so that if a person wants to learn something before contributing, he can just go. There are a bunch of resources where he can go and study them, and like it will help them to like learn the things quickly.
Good. Yeah, so that's very good. Yeah, and like I said, you know, we can like have like a list of URLs for these things, but I guess it'd be in the main. So I have the DevoZoo. So it's DevoWorm GitHub.io, and then it's DevoZoo index and DevoWorm AI index, correct?
Yes.
Okay, so those are the URLs. So that's where everything's going to be hosted. We have, so it's going to be nice. And then I think the only thing left is, I think when this is done, we're going to have to think about maybe how to, more of a community point, but like how to guide people through. Because I know we're trying to make it accessible, but you know, I mean, I think about like people starting to use it and how people are going to be at different levels of expertise with, you know, both like, you know, analysis.
Keeping that in mind, like in the image segmentation notebook that I'm preparing, I'm trying to write down everything that is there involved, like including the time that I got to train the model by using how much GB of CPU, and how to, like, even I'm stating how to turn on your GPU if you are not getting a result. So I'm trying to be as elaborative as I can in that image segmentation stuff.
Yeah, I think that's good. Yeah, and I think like, you know, that sort of thing, and then, you know, we'll have, I think, you know, after we start getting contributors, we might end up sort of getting like these little guideposts where they can go through, you know, they can walk through the platform and, you know, say they want to do some sort of analysis. I think another step might be to have, you know, like maybe videos where people walk through. I mean, this is after GSoC, of course, but I'm just thinking about how we can make it easy to use. And again, it's all going to come down to who's using it and kind of like getting a sense of who's contributing or who's just using it or how it's being used. So, and again, like I said, I think that's good. I think there's always room for like contributing in terms of like data science content. So like if people have ideas about how they might apply, you know, some sort of technique or maybe a tutorial on that, that's always useful. Or, you know, we can go in the direction of modeling, which we don't have a repo up there for yet. But, you know, to have like think about this in terms of models, we can take the images, get data, quantitative data out, and then maybe not a machine learning model, but like a physics-based model or something where we can actually look at different sets of questions. But it's all in that sort of platform. So, okay. So thank you, Ujjwal, Mayukh, and Dick for presenting. And yeah, everyone have a good week. And we'll catch you on Slack if you have any questions. And Ujjwal and Mayukh, I'm looking forward to maybe having a 15 to 20 minute presentation next week in PowerPoint. If that's, let me know if you have any problems.
Yeah, I can do that.
Okay. All right.
We can like share the PPT with you before like presenting it to get reviews.
Yeah. Okay. All right. Thanks. Have a good week, everyone.